All living organisms have genes, inherited units encoded by DNA that allow individuals to transmit information about their traits along to their offspring. We've known that genes exist for over a century. The structure of DNA was discovered in 1953. We've had a complete sequence of a human genome since about 2001. So you might think we should know by now, how many genes do we humans actually have exactly? And the answer is we don't have a precise number, and we never really will. And that's largely because it's hard to define what a gene is in the first place. So before we can decide to try to count our genes, we have to decide on what a definition of a gene is. And that's what this video is going to be about. The definition of a gene is a bit like the debate astronomers have had over whether Pluto is a planet or not. And it has several things in common with that controversy. One is that a definition is thought by scientists to be important, but that definition has largely changed over time, and even still today, it's hard to get everybody to agree on exactly what the best definition is. Gregor Mendel was the first person to deduce that differences in heritable traits were controlled by units that he called elements, that often followed predictable rules of inheritance when they're passed from parents to their offspring. So for example, two forms of elements existed that control pea plant height, either tall or short. If a plant has two tall elements, the plant's tall. If the plant has two short elements, it's short. But if it has one tall and one short element, it's still tall. Nevertheless, the short height element is still there in the hybrid plant. The hybrid plant can potentially pass this short element along to its progeny, potentially giving short offspring. Mendel used dominant and recessive to describe the different effects of these elements. The reliable predictability of these inheritance patterns in the pea plants he studied was powerful evidence that inherited units were real entities, but he didn't call them genes. That term would not be coined for another 45 years. The significance of Mendel's incredible 1865 paper on pea plant inheritance was largely overlooked by the scientific community until about 45 years later in 1900, after his death, when several researchers simultaneously rediscovered his basic observations. The significance of Mendel's work was immediately recognized. And soon, elements following the same kinds of rules of inheritance were found in animals as well as plants. William Bateson named this new scientific field genetics, and then in 1905, Wilhelm Johannsen coined the term gene to indicate these units of heredity. Bateson also created the term allelomorphs to designate the different forms of a gene controlling the same trait. This was eventually shortened to alleles. So Mendel's pea plants carry two alleles for height, either two tall alleles, two short alleles, or one allele of each type. And notice that so far genes could only be deduced to exist if there was observable variation in a trait. In the early 20th century, Thomas Hunt Morgan and his laboratory members at Columbia University used fruit flies to study genes. For example, genes that controlled eye color or wing shape. But they also showed that genes were somehow contained in nuclear chromosomes, thread-like structures in the nuclei of cells, and that discrete genes were organized in a reproducible linear fashion along the chromosomes. Now this fit nicely with new advances in cytology that showed that plants and animals usually had multiple pairs of chromosomes, but that gametes only carried one chromosome from each pair. So it fit perfectly with Mendelian inheritance. Morgan's group even discovered how to estimate the relative position of individual genes along a chromosome, creating the first gene maps. And this led to the idea that genes were arranged on a chromosome like beads on a string. It was also becoming clear that most individuals within a species shared the same basic organization of genes along their chromosomes. But it was still unclear what exactly genes were made out of. However, it was still unclear what exactly genes were made of and how they worked. Definition 1. Genes are heritable units located on chromosomes that control specific biological traits. In 1941, using a simple bread mold, Beadle and Tatum showed how some genes seem to have a simple job. They allow the organism to synthesize one specific type of protein. In their case, the proteins were enzymes that helped create nutrients that the mold needed in order to grow. This idea evolved into what is often called the one gene, one enzyme hypothesis. And since many important proteins are not enzymes, it's more generally useful to think of this as the one gene, one protein hypothesis. Definition 2. Genes provide instructions for making individual proteins. This turns out to be a pretty useful definition. It's still not quite perfect, though. By the 1940s, some observations defied the nuclear beads on a string, one gene, one protein model. Around this time, Barbara McClintock discovered mobile genetic elements that could move from positions on one chromosome to another position on the same chromosome or on another chromosome altogether. Also, some researchers hypothesized that the cytoplasm of cells also contained some genetic information separate from the nuclear information. Both of these ideas turned out to be correct. Not only that, scientists still didn't know what genes were made of. Chromosomes were known to contain both proteins and nucleic acids, but since proteins are more chemically complicated than nucleic acids, most scientists believed that the hereditary material must be encoded in protein. Then in 1944, Oswald Avery and colleagues used bacteria to show that it really was nucleic acid and not protein that genes were made of. And this was confirmed later by Hershey and Chase in 1953. Finally, also in 1953, Watson and Crick described the structure of the double helix of DNA. Now, they relied on crucial data generated by Rosalind Franklin, Raymond Gosling, and Maurice Wilkins. It was immediately clear from the structure of DNA, the double helix, that DNA really could be complicated in that the sequence of bases along a DNA strand could be infinitely complex, yet the strand could be easily copied, replicated, to make new copies of genetic material. 6 years later, Francis Crick famously proposed that the main purpose of DNA is that the sequence of its bases encodes the sequence of amino acids in individual proteins. He called this idea the central dogma. Soon it became clear that in order for the DNA code to be translated into a protein molecule, it first has to be copied into an intermediate type of nucleic acid, messenger RNA, or mRNA for short. Definition 2 revised. Genes provide instructions for encoding RNA molecules which are then translated to make individual proteins. But more discoveries challenged this simple definition. Since scientists can now directly compare DNA sequences with gene maps, a few surprises emerged. One such surprise was that certain genes actually did not seem to encode proteins at all. Research in bacteria had clearly implicated that some segments of DNA didn't encode protein, but were crucial for controlling the step in which the RNA intermediate was made. This step is called transcription. In bacteria and viruses, most of their DNA does in fact encode protein, but in plants and animals, most of their genomic DNA is this non-protein coding stuff. Some of this extra genomic material in animals and plants is devoted to the transcriptional control of nearby genes. For example, Ed Lewis carefully mapped a series of distinct genes that control development of fruit fly body segments, but some of these genes turned out to be these sorts of regulatory sequences. Furthermore, in 1977, it was shown that mRNAs made by adenovirus, which infects human cells, were often generated by splicing together bits from much longer RNA molecules. The intervening bits of RNA get quickly recycled by the cell. Amazingly, it was soon shown that most animal and plant genes are spliced from these longer RNA precursors. The bits that get retained in the mature messenger RNA were termed exons, and the bits that are spliced out were called introns. And not only do most human genes go through this splicing process before translation, for almost all human genes, more of the RNA is spliced out and recycled than is left in before translation occurs. So most of our genes contain big stretches of DNA sequence that don't wind up encoding any protein at all. Finally, for some very important genes, they don't encode a protein, but they do encode an important functional RNA. Sometimes the RNA molecule itself is the functional end product. Ribosomal RNAs and transfer RNAs are in this category. They're functional in their own right, playing key structural roles in protein translation. Lots more examples of functional RNA genes are now known. However, it's generally more difficult to infer the function of an RNA molecule from its sequence alone than it is for a protein sequence. Definition 3. A gene is a portion of a DNA molecule that encodes an RNA that is translated into a protein, except when it isn't, and even if it is translated into a protein, a lot of the RNA might be thrown away first. Notice how at this point, our definition of a gene has lost sight entirely in describing genes as actually affecting any visible phenotype of the organism itself. It's purely a molecular definition. But to count genes, this molecular definition is really important and more useful than the phenotypic definition, because ultimately we care about the DNA molecule itself, and especially mutations in these molecules and how they affect human health. Here's a modern definition of a gene based on a 2007 paper in Genome Research that was devoted to just this problem of gene definition. A gene is a union of genomic sequences encoding a coherent set of potentially overlapping functional products. This is a good example of how tough it is to define a gene precisely and concisely. And notice this definition doesn't even include the words RNA or protein, and it does include the phrase functional. Now, most people would agree that a gene has to do something. It has to have some sort of recognizable function. One problem is that it can be easier to show that a gene produces a related RNA or protein than what the function of that RNA or protein really is. And a further complication is alternative splicing. Since there's potentially more than one way to splice up an RNA following its initial synthesis, a gene could encode more than one type of functional RNA or protein. But using the previous definition I just cited, we'd still count this as one gene. The goal of the GenCode project is to create high-quality annotation of human genes, and it's a leading authority on the human gene count. Using a reference human genome sequence as its guide, the project currently recognizes 19,955 protein coding genes in the human genome. But it also counts about 25,000 RNA genes that don't seem to encode proteins. The majority of these RNA genes are of unknown function. In fact, some of them may be transcriptional background noise. Because the human transcriptional machinery is so complex and the genome is so big, we can tolerate some extra RNA production here and there, especially given that so much RNA is immediately recycled after synthesis anyway. But we really don't know how many of these RNA genes are functionally important, as most of them have not been well studied. And we haven't even talked about pseudogenes, imperfect extra copies or fragments of copies of other genes. These are scattered across our genome. We've got thousands of them. And most of them are byproducts of ancient mutational events that duplicated chunks of our ancestral DNA. They're usually easily identifiable as non-functional broken bits, but for some pseudogenes, it's hard to know if they might still retain some function. But we'll have to wait to talk more about pseudogenes in another video. Finally, all humans have small differences in the amount of DNA that they carry in their cells. So I might be missing a few copies of genes that you have, and vice versa. Your gene count or mine might be slightly different than the official GenCode count. It's still not likely to be very different. I'd guess less than a fraction of a single percentage point. But these differences may be part of the stuff that makes each of us unique.